Good afternoon, everyone. First of all, thank you to Dr. Harsha, Dr. Reena, Dr. Nisha for having me here today. I'm really grateful. And whilst we are on the topic of diagnosis, it's extremely important to think about costs. And not only that, which diseases that you're diagnosing, because sometimes diagnosing diseases that are ultra rare can become extremely challenging and can be a costly affair. Now, before I begin, I just want to highlight a few conflicts of interest. I'm a named inventor on two patents. One is international, one is national, describing technologies that are used in detection of rare diseases. And the second one is that I spearhead Fridge and Sanofi Disha program for free lysosomal storage disorder diagnosis in India. Now, I have been involved in a whole host of things in the past 10 years, starting from the pharmacogenetics of warfarin to rapid molecular diagnostics with a spin-out company. Well, now it's rather a huge firm called Quantum DX in the UK, to studying male infertility, to studying aspirin-based chemoprevention in Lynch syndrome. But it's only after coming back to India in 2019, I realized there are a huge number of challenges that are currently facing Indian community, starting from lysosomal storage disorder, autism spectrum disorder, male infertility, and not only that, binding into all together is computational power, because to understand all of these things, you require a whole host of computational knowledge and grunt. And that is exactly what I lead now. I lead the computational biology core unit, and I also established Advanced Genomic Tech Division. And the whole energy behind leading all of this is actually summarized in this one line that was written by Thomas Adams in 1618, that he is a better physician that keeps diseases off us than that cures them being on us. Prevention is so much better than healing because it saves the labor of being sick. And this is absolutely true when it comes to genetics, because this is one of the few branches of medicine that allows us to prevent the diseases from coming in the very first place. So today, my focus is purely going to be on lysosomal storage disorders. And you might already know about this quite well. There are a group of 70 different disorders put together, and they follow a recessive or an X-linked inheritance models. You have a systemic collection of phenotypes that means it's not only just one organ, but a whole host of different organs that are affected. And the combined prevalence worldwide is about 1 in 1,500 people, 1 in 5,000 people. So that's quite common when you put them in those numbers. Not only that, when you think about India, out of those 70 different diseases, there are 20 odd diseases that are extremely common. And this work was actually led first by my father in 2014, nearly a decade ago now, Dr. Jayesh Sheth. And he was the first one to show that Gaucher's disease is, in fact, the most common lysosomal storage disease in India, followed by Tay-Sachs and Sandhoff disease. Now, when it comes to diagnosing these children, the pathways, well, for the past 2 decades has been rather simple. You see the child in the clinic, you try and understand the symptoms and perform a clinical differential diagnosis. You would then do a biochemical screening or a diagnostic test to identify in which group does this child belong to. Then you would do an enzymatic confirmation to be absolutely sure that, yes, in fact, you're dealing with Gaucher's or MPS type 1 or type 2 or whatever it may be. And once you have that, you then perform a sequencing-based test or an MLP or some sort of genetic test to really identify the cause as to why this child is having this particular disease. The problem is that this is rather iterative in nature. So if you think about mucopolysaccharidosis, you've got multiple types of MPS disorders, and they all have overlapping phenotypic symptoms. So if you come across a child with MPS, it can become extremely tedious because you have to go around the circle multiple times. And because of that, sometimes the diagnostic odyssey can last up to 5 years. The per patient cost increases quite dramatically because you're going around the same circle again and again. And each time you go, it requires skilled manpower. And because it's skilled, you require skilled manpower in a complex lab setup, the cost starts to spike. And because of that, it is estimated that the diagnostic yield for LSDs in Belgium and even in India is all the way down to 5%. So if I put that in raw numbers, there are estimated 240,000 kids in India with LSDs right now, but only 12,000 of them are known to medical services. So that's a whopping number. And therefore, we require a high throughput, low cost, and easy to use diagnostic assay to identify these kids. Now, as a geneticist, my immediate reaction is, well, let's just do sequencing on anything and everything that moves. But then there's a problem. If you try and sequence everything there is, so you would see the number of genes at the top, and you would see the detection rate at the bottom. No matter how many genes you had, the highest you could aim for was about 65% diagnostic yield. That's not great in my view. I think as a geneticist, we can perform much better. Now, the other problem with NGS is the more genes you add in a panel, the cost starts to spike up. And it's not because the panel size has increased, but the laboratory setup you require also becomes bigger. The computational tools become bigger. The manpower required also becomes large. So if you want to make a panel, especially for a country like India that is cost sensitive, you need a panel that is small, focused, and easy to deploy. And that is why when I started to design a panel for LSD, I thought of Illumina MiSeq. That's the smallest instrument there is in the Illumina sequencing setup. And it's present in majority of the labs. And not only that, I focused on just 29 diseases that I know are common in India. So here are the list of diseases that I started to work with. And of course, this was the first father-son duo project that I had the pleasure of working with. And of course, I have my student, Adira Nair, and two colleagues from GBRC, Dr. Madhvi Joshi and Professor C.G. Joshi. And to cover these 29 diseases, we had 23 genes. And the panel was designed in accordance with American College of Medical Genetics sequencing guidelines. The aim was that it should fit well within the Illumina MiSeq platform. It should be able to detect not just single letter differences in the DNA, but it should be able to detect deletions and duplications, because in certain diseases, they are quite common and NGS has a tendency to miss them out if you're not careful. And most importantly, it should be cheap and it should be amenable to high throughput. And to do that, I went back to a technology that was first described in 1994. Back then, it was known as padlock probes. Today, it's known as molecular inversion probes. And to summarize it rather simply, think about a DNA molecule giving your target region a big, nice hug. That's exactly what it's doing. And every time it gives a hug, it makes a copy of the region that you're interested in. And it gives it a barcode. And when it gives it a barcode, you're able to identify how many unique DNA molecules you are able to capture. Now, when you're designing these probes, they're rather expensive. Each probe costs about $20 to make. But the beauty is, once you've designed it and you've manufactured it, they last for more than 1 million reactions. Or to put it in simple terms, they cost less than 2 cents per sample. So they're extremely cheap. And the first time I had used them was for microsatellite instability testing in colorectal cancer patients, which I'm very happy. There is now an international patent covering it, and it's being deployed in the UK's National Health Service. The pathway is very, very simple, just like any other genetic testing lab. You do a preparation of your sample. You would pool how many samples you have together. You would sequence them. You would run a computational analysis, and you have a result at the very end. And I'll break down these parts very, very simply for you. Once I started to design this assay, the first thing I wanted to check was, is it doing what it is supposed to be doing? So I had 49 samples where I already had the mutation known. And I said, okay, let's try out my assay and see whether it's able to capture that. And lo and behold, got 100% concordance, which is great. But what I hadn't anticipated was how good it really was at capturing deletions and duplications. So here is an example of a patient with a mutation in GALC gene. And you have nearly 4 to 5 exons that are deleted. And they're heterozygous. It means the deletion is only in one copy of the DNA. The second copy is intact. So it was excellent at picking that out. But then I started to come across samples where just one exon, just a very small part of the DNA was deleted. And lo and behold, the assay was able to capture that. And this meant that there was no longer a need to do MLPA-based testing. I could capture all of this in one shot. So over the next 2 years, me and my father embarked on a journey to recruit 250 odd patients. And to date, we have recruited 259. We did testing in all of them. And the idea was that these are the patients where either only biochemical test is done, or a doctor suspects that this patient has an LSD, but doesn't know which one. So in this cohort, we got a diagnostic yield of whopping 83%. If I have to split it based on the type of patients we had, if they only had a biochemical testing done, our yield was up to 87%. If a doctor just suspected that this patient has an LSD, our yield was 74%, because some of these patients had an LSD, but it was a very, very rare form that wasn't covered by our panel. Now, here's the part that I'm most proud of amongst all of this. In Gaucher's disease patient, you have one particular mutation that is most common. It's PL483P. It's a rather boring designation, but essentially, you find it in about 40 to 50% of Gaucher's disease patient. And even if you were to do whole exome sequencing, you'll not capture it, because the part of the DNA where this particular mutation is present looks eerily similar to another part of a pseudogene, right? So whenever you do sequencing, it tends to miss things out. But our assay is able to pick it up with 100% accuracy. So we are better than exome, at least for this point. The problem is that even for that in Hunter's disease, the same problem creeps up. And this is where our yield is actually going down. That is why we are not at 100%, we are at 87%. But lo and behold, this is still better than any other assay that is out there right now. And to cover all of this work, we now have an Indian patent. In terms of cost, because we are talking about a very cost-sensitive market, when you have a patient coming through the clinic, the cost of counseling, DNA extraction, sequencing, reporting, and post-test counseling, we currently do it for $120 per patient. This is the cost to the patient that we have right now. If we get a variant of uncertain significance, that means we don't know whether this mutation is causative or not, and we need to perform a biochemical test, it's just $20 extra to the patient. So the cost is $145 at this point. Now, because of the instrument that we are using, we have the flexibility. We can have as low as 4 samples, or we can have as high as 50 samples in a single run. And the cost remains the same. The data analysis time is 30 to 45 minutes. It means I can run the analysis on my laptop, by the way, in under an hour per sample. I don't need a fancy big computer or a server to do this. My laptop is good enough. And the overall turnaround time for 50 samples is less than a month. So should I just say that my assay is the best thing there ever was and you guys should adopt it? No, I don't think so. If a doctor is absolutely sure that this patient has a Gaucher's disease or Hunter's disease or Hurler disease, go for the traditional approach, because the cost of biochemical test is about $30 to $40, and you get to the diagnosis within a few days. But if you are not sure as to which LSD this patient is having, then go for this new approach, whereby you first do this particular assay, and based upon what variant you find or what mutation you find in a gene, you then do a biochemical confirmation, and you get to that answer. Now, whilst this assay is currently being panned out, I'm very happy to say that right now, we are funded by the Department of Biotechnology, which is an arm of Government of India, to create India's first biobank for lysosomal storage disorders. And this includes all 29 disorders that I just showed you earlier. And in this, we'll have DNA, plasma or serum, urine sample, biochemical and longitudinal clinical data, and anthropometric data for every single patient. Each patient will receive a biochemical test and a genetic test free of cost. So this particular assay will be utilized in this work, and the data will be made freely available, anonymized, starting from 2024. Again, this is a father-son duo work, so I'm very proud of that. And when it comes to clinical testing, I'm very, very happy to share that this assay is now out in clinical utility. Sanofi, which is the world's biggest drug maker for LSD, partnered up with us to create what is known as Disha program. So there are 5 LSDs: Pompe, Gaucher's, MPS1, Fabry, and ASMD. They are doing this diagnosis free of cost to the patient. So this includes both biochemical as well as genetic diagnosis. And we have so far done this on more than 100 patients. And we are very, very proud of that. In conclusion, even though this biochemical test is the current gold standard technique, it can be iterative, not amenable to high throughput, and can be rather costly. Our assay is able to cover 29 diseases, LSDs that are common to India, and is able to detect both point mutation as well as large deletions and duplications with a whopping 83% diagnostic yield. It is low cost, it is high throughput, it is accurate, and has a quicker turnaround time. It has, because of its abilities, it is flexible in deployment right now, and the assay is available in clinics right now. To do all of this work, of course, it's not just me and my father. We have a wonderful team right behind us. We have got wonderful team members, and as you can see, mostly are females, very few males, and all the wonderful national collaborators out here, including Dr. Cabra, and our funders, GSBTM, Department of Biotechnology. And I'm, again, very, very proud to say all of this is done, made in India and for India. Thank you.